Hello, everyone. Welcome to Hudson River Park's Ask a Scientist. This is the first in a virtual summer lecture series that will allow you to hear from local New York City researchers and experts and get a chance to ask them questions, both about their work and also the science that they do in general. We at Hudson River Park's River Project collaborate with dozens of scientists from year to year and season to season from all over the city and neighboring areas. And this year we are continuing our partnerships the best way we can in a primarily virtual season. So thank you for joining us today. And without further ado, let me introduce today's speaker. Dr. Peter J. Park of Farmingdale State College, formerly of Nyack College, is an ichthyologist who was born and grew up in Queens, New York. His expertise in evolutionary ichthyology with current research interests in fish cognitive ecology, fish community science, aquaponics, phage biology, and teaching science through active learning. He is also an avid angler and overall fish enthusiast. So please help me welcome Dr. Peter J. Park. Dr. Park, thank you for joining us today.
Thank you, Roberta. It's a pleasure. Pleasure's all mine.
Well, I'm gonna turn it over to you, and we're really looking forward to seeing what you put together for us today.
Thank you. Okay. Well, thank you everyone out there. I can't see you, but I can sense you. My name is Peter Park, and it's really a privilege to give this lecture and have the opportunity to answer questions that you might have. I also want to thank Hudson River Park and Lamont Doherty and the River Project for giving me the privilege to give this talk. It's not every day I get to share not just about my work, but about my personal story as well. And I also my name is Peter. Good? Are we good?
Yep.
Okay.
Yeah, we're good.
Thank you. All right. So without further ado, I will start the talk. This is the outline of my talk this afternoon. As you can see up here, I have a question. I can't believe I have the opportunity to give a talk like this. I'm a scientist. I'm going to share a little bit about my childhood. Also briefly describe some of the PhD research that I did, and then share with you something that is more than just a hobby to me, fishing, followed by some current research projects and outreach work that I've had the amazing opportunity to do, and some additional education projects. And I'm going to conclude with some cliche type of life lessons that I've learned throughout my journey. All right, so this next slide, it's a little embarrassing, but worth it. So that's me when I was about, I think, 10 years old. My father used to take me fishing a lot. As you can see, as a very young kid, I really enjoyed fishing. And I grew up here. This is Sunnyside. I was born in Flushing, but I grew up in Sunnyside. And just some landmarks. This is the 46th Street Bliss Station for the 7 train, the Sunnyside Arch. And wanted to acknowledge a really important pet store that allowed me to really hone a lot of my interest in fish, because as you can see, I grew up in the city. And so there was nature, but not sort of the picturesque scenic nature that I often think about when I think of searching for fish. So as a young child, I actually took care of a lot of fish as pets. And thank you to Pets Unlimited for enabling that and making that happen. Growing up, this is where I lived. I don't live there anymore, but this is where I lived in Sunnyside. And this is 48th Street right here. And this square shows you what my parents did. My parents, they owned a fish market. And so probably starting to put the pieces together. I'd like to go fishing. Parents owned a fish market. And I want to share this little blue square here because some of my earliest memories take place in this corner. Why? Because whenever it rained, this corner used to flood. And I would always ask my parents or my grandmother if I could walk outside and just literally stare at the water, hoping that fish might emerge from that pool of water that I know would be there after rain. Never happened, but you know, that was sort of my imagination running wild as a child. So today, when someone asks me, you know, who I am as a scientist, three qualities kind of jump out at me, and I share them here. So my background, my ethnic background, I'm a Korean American. My parents are from South Korea. They immigrated here, and I was born here in the United States. My parents, for as long as I lived, had a fish market. My mother still has a fish market to this day. Her market is in the Bronx now. And of course, fish. And so there's something about this animal that just entrances me. I just think they're fascinating for so many reasons. And I'm just gonna stop there because I could go on forever about why I think fish are so cool. So that's a little bit about me. Let me share a little bit about my research during my time at Stony Brook University, where I did my PhD. I studied a little fish called the three-spined stickleback, Gasterosteus aculeates. And they're found all over the Northern Hemisphere, but they're very abundant in areas like Alaska. And so this is Alaska, this is South Central Alaska, which is where I focused much of my work. And what's really special about this fish is that it's like salmon, not related, but like salmon, they're sea-run, meaning that they live out in the ocean. And when it's time to breed, they come into fresh water. So usually, you know, those are streams, rivers, lakes. And what's neat about this in areas like South Central Alaska is that there are hundreds of different kinds of lakes. And many times these populations that go there to breed get landlocked, and the populations adapt. And so there are what we call resident freshwater populations that haven't had a chance to go back out into the ocean, and they live generation after generation in the lakes, and they adapt, they evolve. The particular ecological dichotomy that I focused my work on was the shallow lake and deep lake dichotomy. So in a shallow lake, what makes this lake dichotomy interesting is in shallow lakes, typically you have highly structurally complex habitat because plant growth is permitted throughout the entire area of the lake. And much of the prey that's present there that the stickleback forage on are relatively large benthic invertebrates. In contrast, in deep lakes, you have a lot of deep water, too deep for plants to grow, and the predominant prey is plankton that's swimming in the water column. And in addition, I mentioned that these fish adapt, but it's really striking and visible. For example, if you look at their body shape, the shallow lake stickleback tend to be very muscular, and that helps to maneuver in those tight spaces of their complex habitat. And those that live in the deep lake, the freshwater populations there, they're built to cruise, so they tend to be more elongate. So if you know your fish, you probably recognize this. Like when you think about open water fish, like swordfish, they tend to be very elongate, like mackerel, tuna. Meanwhile, fish that are structure oriented, like wrasse or scup, they tend to be very thick bodied, which helps them maneuver into tight spaces. So this is the context from which I started my work. And so I was particularly interested in brain and behavior, specifically spatial cognition and the forebrain. So those were the areas of interest that I focused my dissertation on. And so I guess if I were to ask simply, comparing populations that are shallow versus deep, who are the better learners? Learners in the sense of better spatial learners, ability to navigate in a complex habitat, and who might have, if there are differences in brains, bigger brains. Not saying that bigger brains are better, but just saying that it'd be interesting to see if there are differences in neuroanatomy. And so right off the bat, if you can remember one thing, I highlighted this sentence up top there. If you think that a fish has a 3-second memory, I'm going to debunk that myth right now. they don't, and I'm going to show you some compelling evidence to show you that that is the case, that they have long memories. In order to explore spatial cognition, I implemented an experiment called a T-maze, which is a standard maze pioneered with rodents in the field of psychology, and has since been adapted to fish. And so basically the way this works, and I'm going to show you a video in a second of a fish swimming through a T-maze. It's called a T-maze because it's shaped like a T. At the base of the T is the start site, and one of the arms are, at the end of one of the arms is a reward. In all of my experiments, the award was food. And fish in general didn't get to see the maze. Well, there was about a 24 to 48 hour period between exposure to the maze, and I implemented 2 trials per day, and then again 2 trials per day, and then about 24 to 48 hour gap in between the 2 trials. So the fish are only getting fed in the maze, and so they learn to associate the maze with feeding. These are 2 videos of the same fish. Trial number 1, for the fish is first getting exposed to the maze. So in this environment, in this moment, this fish does not know what it's in the maze for. This is trial 35. So many consecutive trials. And I'm going to play them both at the same time. Okay, and just to show you, well, I'll let the video speak for itself. So that fish at trial 35 is already feeding. I'm going to pause this here. Fast forwarding, you get the idea. So fish can absolutely learn. And I use this experiment with a number of shallow lake freshwater populations of stickleback and compared them to deep water lake stickleback. And this is a figure, it's a plot of the results. Each dot represents an average. Each N represents sample size. So you could see sample sizes for the different populations, which are shown here on the x-axis, were ranged between 11 fish to 16 fish. And on the y-axis, you have here number of trials to reach mastery. So there was a criterion that was implemented, and basically a lower score meant the fish learned the task sooner. And statistically, the shallow lake fish actually outperformed the deep lake fish. So the shallow lake fish are in red, and deep lake fish are in blue. And so who are the better spatial learners? Turns out that the shallow lake fish are the ones that occupy the spatial complex habitat. And this work has been since been validated in other populations outside of Alaska using stickleback again. It's also interested in brains. And so in the literature, we find that in fishes, it seems that much of spatial cognition is processed in the forebrain. This is a dorsal view of a stickleback. These are forebrain lobes, optic tecta, cerebellum. You have the hindbrain back here and olfactory bulbs up here in the front. This is the location of the forebrain. And literature reviews cite multiple times among fish species, those that live in more spatially complex habitats tended to have larger forebrains. Now, what that means functionally in terms of behavior, we do not know. There's a lot of steps involved between behavior and the actual brain work. There's a lot of work to do in between, but this is a good investigation to explore. This is a picture of a shallow lake stickleback deep-bodied, deep lake stickleback very thin. Notice that one thing that was really striking in the work that we did here was the brains look different. And this is fascinating because these are populations of the same species. And prior to the work that I did, much of the work that was in the literature was comparing individual species to other species. So without going into too much detail, you could read more about this work from the titles below. And just email me, I'd be happy to share the papers with you. In conclusion, what we found is that shallow lake stickleback, which live in spatially complex habitats, tended to have larger and in shape-wise actually had rounder forebrains. And so there's much more to the story than just the forebrain, which I will just mention now that there's, again, a lot of more work to be done. But what was really special about this work, I believe, and we believe, my collaborators and I, is that this was something that we documented within the level of a species. And so across populations within one species. All right, so that's research. That's definitely the longest part of my talk. remember that picture of a kid holding a fish? Yep, that's me. And those of you who know me know that fishing is more than just a hobby to me. It's something I continue to do. I have a few pictures of memorable fish here, some New York City fish, and some fish that I've traveled to catch. So I'm just going to leave it there because it's going to provide context for the next portion of this talk. So current research, really exciting research to share that relates to ecological patterns and community science and local partners. And at the outright, the project is informally being called the East River Fish Project New York City, and I want to acknowledge my collaborators who are also very good friends of mine. Isa Del Bello, she is the education director at Brooklyn Bridge Park Conservancy, and Chris Dragenti, he is the natural areas manager at Randall's Island Park Alliance. And the three of us teamed up together and we wrote a grant. And we got it. In 2019, we got a small grant from the Marine and Coastal District Conservation Education and Research Project Small Grant Program. And through this grant, developed the beginnings of a partnership of three that has then expanded to many more organizations. And with this grant, we actually had three goals in mind. One, to develop an East River fish database. Two, to develop new fishing clinics, which can double as an experience to learn how to fish, but also allow the public to collect data alongside us and with us and for us. And then we also implemented, and still in the process of doing this as well, fishing workshops. So originally we had intended to have these fishing workshops in the winter, but with everything that had occurred with COVID-19, we had to cancel two of the three that we had planned. And so stay tuned, possibly more on that in the future. So the fishing workshops were planned to be off season in the winter and to encourage people to learn and hone skills to prepare for the following season. One thing that the three of us realized that was just so amazing and inspiring was we came to appreciate and just be in awe of the fact that along the East River, there were at least 9 organizations in total that were already collecting fish data. And they have since become partners and primary data contributors to the database aspect of this project. And so you can see they're listed here. And I want to acknowledge them and just thank them for all the effort that they did last year to share their data. And we did our best to standardize the way we collected data and talk about the data and improve data quality. And so there's so much here on this slide that is very meaningful to us. And so just thank you if you're out there listening. One thing I also will mention, informally we call ourselves the East River Ichthyological Alliance, or AREA for short, because it's a mouthful to say all of the 9 organizations here by name. But I just want to, so you'll see this term elsewhere in the presentation, but just sharing with you that when I use those 4 letters, it refers to this group of nine outstanding organizations. We also recruited anglers. So remember, I'm a fisherman and I've been fishing. I grew up fishing first before I thought about becoming a scientist. And so these are friends new and old, who I trusted to share data with us. Sometimes, fishermen can sometimes rarely, exaggerate a catch. Everyone that contributed data as anglers were very good about documenting, photographing with scale, and providing additional information to validate the quality of the data that they were being contributed. I also want to thank for hire fishing operations that operate along the East River and in and around the East River, but also fish in the East River, and also fishing clubs that came out, and many of the fishing clubs actually assisted in our fishing clinics last year, excuse me. And we're also able to find additional data points from social networks. So just kind of scanning what people are posting, following up, and generally everyone we contacted after we found a really cool fishing picture, they were very nice and kind and shared additional information, whether it was from iNaturalist, Facebook, or Instagram. So the data that I'm about to share with you, in brief, really brings together a variety of communities that all have one thing in common. They all love fish. And much of this work has been, the data that's being collected is in the form of an Excel spreadsheet, but part of the grant was to build a database and build a database that's going to be accessible and online. And we're calling this this is coming soon. So there is data, there's data sitting in a spreadsheet, but it's also being formatted in a way that's going to be computer-based. And so coming soon, there is an online East River fish database. And so stay tuned for that. It takes a lot to kind of, pull back and not share everything about this interesting sort of direction that the database work is going in. But I will tell you, just stay tuned. And I want to also acknowledge my good friend and super talented software developer, Andrew Hong, who's been the primary person who's been constructing this project, the database element project of this work. So that's the database stuff. What did we find? So which fishes did we find in the East River in 2019? To kind of lay the context for this, I show you a trophic pyramid, a super simplified trophic pyramid, where you have phytoplankton, zooplankton, small fish that eat the plankton, and then large fish that eat the small fish. And there are larger fish that eat the large fish and so on. So this is super simplified. But I show this to you because it provides valuable context for how the photos I'm about to show kind of make sense. So forage fish, those small fish, who were the primary small fish in the East River? Atlantic silverside, Atlantic menhaden, striped killifish, mummichog, and the striped anchovy. So those were very common forage fish that were identified from the data that were contributed by all of our partners. Large fish, who were the large fish? And coincidentally, many of the large fish were contributed by our anglers and for our fishing operations. And these are just, again, coincidentally happen to be sport fish. And so you have striped bass, bluefish, black sea bass, oyster toadfish, summer flounder, scup, tautog, cunner, and orbogal, and the striped sea robin. So these were common catches on the larger end. I also have a slide here, what I call notable catches. And so notable catches are catches that have a story. So it was really cool to see a nearly 3-foot American eel caught. It was really cool to learn that in the East River there were really big summer flounder caught, a very big scup caught. We saw pufferfish, and we also saw a conger eel, which is typically a deep water species. And fishermen and anglers, fishermen probably may not be familiar with these fish. This is a feather blenny, naked goby, and a skillet fish, and a spotted hake. Those are cool fish that were found in the East River last year. And also probably the biggest fish that was documented was this nearly 25-pound striped bass in November. We also got some tropical strays. So these fish are visitors. They are strays that have been swept to the Gulf Stream from southern waters. And last year we documented 3, a butterfly fish, a gray snapper, and cobia. So how well do you think you know our East River? I have a question for you. In 2019, how many total species of fish do you think we caught in the East River? 47. And we documented a variety of techniques and how fish were captured, whether it was seining, a large 15 or 30-foot net, cast netting, dip netting, trapping, or angling. Question number 2, does catch method matter for research? Absolutely. And to kind of highlight this is a snapshot of the database showing you on the left, you have fish species, on the right, you have catch method. I highlight here fish that were caught exclusively using one method, and note the diversity of the methods that are shown there. So for example, the gizzard shad was only caught with a cast net, the conger eel was only caught via angling. And so does a variety of methods matter when you're trying to document what's swimming in the East River? Absolutely, that's what our work showed. And this graph highlights that aspect a little bit more. So if we're just looking at fish species and disregarding abundance for the moment, nearly 50% of fish were caught via net and 50% were caught through other approaches and methods, and nearly a quarter of fish species were caught via angling. All right, so so that concludes sort of the major research elements of my work. And I now want to share very briefly some outreach work and educational projects. So for outreach, as part of the MCDL grant from last year, we had resources to initiate and establish fishing clinics in Brooklyn Bridge Park, World's Fair Marina, and Randall's Island Park. Brooklyn Bridge Park and World's Fair Marina were two sites where we had beginner level fishing clinics. And so this beginner level clinics, meaning clinics where everything was prepared for you, from handling fish to even sometimes having photos taken to handling bait and so on. Everything was pre-prepared. In Randall's Island, we tried, and I think we were successful, implementing what we call the intermediate level fishing clinics, where this was a more DIY approach, where we had a tremendous support from local tackle shops like Jack's Bait and Tackle in City Island, where donated fishing supplies, and anglers were encouraged to try to catch fish, and that's it. And if they needed some guidance from experts, we were there on hand. No judgment, just kind of came, and if they wanted to learn how to tie knots, rigs, or just explore something that they thought about, didn't matter, we helped. And if they caught fish, great. If not, didn't matter either. But we caught a fair number of fish through all of our clinics. I also want to acknowledge the Department of Health. Throughout all these clinics, there was literature made available on fish consumption advisories that were translated in multiple languages. And so, as you can see, I'm an Asian American. I grew up fishing in New York City. I want to kind of highlight just briefly my friends who helped put on the World's Fair for World's Fair Marina clinics, of which we had two. What was really special about this is many of us actually, that you see in the photo here, grew up fishing and kind of fishing alone. And it's really neat. As we grew up, we kind of sort of found each other. And also many of us are children of immigrants. We have immigrant parents. And when we put this clinic on, it was really neat to not only have materials that are prepared in our native languages, but also participants. There were several participants that did not speak English, and we were able to accommodate them because we still speak our native language. And so just privileges and opportunities like that were really special memories from 2019. Thanks to that, thanks to the help of our amazing staff volunteers and MCDL grant. And thank you also to Swivets for your generous donations of concrete lead-free sinkers. I also do citizen science, community science work with Macaulay Honors College. And they have this tremendous program where they get students, hundreds of students, out to an iconic park, and they collect data on everything that lives in that park or an identified geographical region that has special significance to New York City. And I've been very fortunate I'm privileged to be their fish person for these events, taking teams of students to identify what swims in the waters in these parks. So I want to acknowledge them. Kelly and Lisa, if you're out there, thank you so much, and looking forward to many more bio blitzes. Educational projects. One fun project that I got to work on in the early days of my career was the Stickleback Evolution Virtual Lab. If you have time and 3 hours to spare, you want a primer on ecology, evolution, paleontology, statistics, Mendelian genetics, molecular genetics, try this virtual lab. It's free, it's online, and if you're an educator, please feel free to contact me if you need any ideas or suggestions on how to implement this in the classroom. But this is a really neat free virtual lab that gets you to Alaska to study not brains and behavior. The work here is primarily the work of my thesis advisor, Dr. Michael Bell, and also the work of Stanford Stickleback biologist, Dr. David Kingsley, who worked with Stickleback, but he works with a tremendous array of model organisms and does really fascinating work that pertain to medicine. Always looking for opportunities to to work with fish. And so I've dabbled a little bit in aquaponics. Aquaponics is the practice of coupling gardening with fish keeping. And the logic here is that fish defecate and they make waste, and that waste can become fertilizer for plants that are connected to that water, and that those plants can, in a sense, in some ways, chemically filter that water as the water trickles back. So you have this really neat, small scale, somewhat sustainable system where you could grow fish and grow a garden at the same time, and it can be done indoors or outdoors. So really fun projects that I've had the opportunity to. This one with collaborators at Duquesne University and Borough of Manhattan Community College, and this work was done at Nyack. So we're near the end. I don't know how I'm doing on time, but I wanted to kind of share with you some life lessons. And before I kind of jump directly into them, I want to share some really cool stories. This is a student of mine, a former student of mine, who was attending a community science program. And we took this photo, and this photo happened to win the 2019 Women Hunt Fish New York Saltwater Division, and it represents a striped bass. It represents students being involved in research in the field, and it represents the East River. We're very happy about that photo. And there are countless stories that I could spend hours sharing with you. This is just a snapshot of some of the stories from last year. So this is my friend Gary. We grew up fishing in the East River since we were really, really young, like 5th grade, 6th grade. And so we still fish the East River together. Students, Nyack College Fishing Club. Also, Brooklyn Bridge Park Conservancy's fishing clinics were featured on the news last year. So shout out to Spectrum News New York 1. Families that I came out to multiple clinics over the years, not just 2019, but other clinics that are on the East River. We even had a fish photo and story contest of area organizations, and this is Randall's Island Park with Jen and Jackie and Chris showing off their first of hopefully many annual the first of hopefully many annual contests for what we call the most significant catch in the East River Award. And just taking friends and family out fishing in our local waters and just opening their eyes to what swims. And that's the Throgs Neck Bridge in the background there. Oops. And one really, really cherished memory of mine from last year is having the privilege to bring together the environmental education community with the for-hire fishing community, bringing our environmental educators on a boat, fishing local waters. And these are fish that were caught on a trip with the Capital Princess and a trip with Never Enough Fishing, who both dock in the East River. And it's my friend Mike there with a really large striped bass caught in November right next to the UN building. And I'm keeping that vague. It's not giving away the exact spot, but it was a pretty cool fish, biggest fish of the year last year. And our friends here, just so many smiles, so many memories. All right, so what are some takeaways that maybe can encourage you and invite you to ask some questions? One, dreams can come true. I'm a Korean kid from Queens. Parents owned a fish market, and I became a fish biologist, and to this day, it humbles me to just reflect on that. Chart your own path, and don't worry if you have unique interests. At the end of the day, if you're passionate about something, people appreciate your passion. Failure, expect it. It's part of the process. Just get back up and press forward. Prepare now for unexpected opportunities and luck later. So prepare now. Advocate for science. Think critically and speak your mind, but also do take time to always listen, because everyone has a story. Get involved, search online, participate, learn, document, share, and like stuff, like the East River Fish Project New York City Facebook page and Instagram account. And also remember that you're a part of a community. So wherever you are in and amongst the community. And one day you will build communities, and also realize that the community you're in were built by others. And there are no shortcuts there. So just acknowledge and appreciate the moments and the friends and the colleagues and collaborators that you have in the moment. And as riveting a lecture as I hope this was, one thing I learned is that people will always remember experiences and relationships first and before any lecture I have ever been able to give. And so just appreciate those moments. And when things get back to some sense of normal and you're able to go outdoors and interact and learn about your East River, I hope and invite you to just cherish that time, and because you will remember those in-person experiences. I want to thank funding sources for the work that I shared in this presentation and our tremendous list of colleagues, collaborators, and partners that made the work in this presentation possible. And with that, I will take questions. Hopefully I did okay with time. I think I went over a lot.
Peter, that was perfect. Thank you so much for that incredible presentation. Your work is so valuable. It's so widely varied, covering so many different topics, and it was really wonderful to see it all laid out there. So thank you very much. We do have quite a few questions already. So I think we're going to transition right into the Q&A session. We'll get started. We have questions from several of the different groups that are watching, internship and related groups watching, as well as some individual people on the Facebook stream. So we'll just get started. This one comes from the Intrepid Goals program. You mentioned in one of your papers that are in the article that fish from the East River seem to be smarter. How can you tell? Have you done any experiments to determine this?
Purely anecdotal. How can we tell? Ask any fisherman who has fished outside of New York and also fishes in New York. The fish in the East River are way harder to catch. And there is two observations I want to share very quickly. One is in science, we typically, I wouldn't say favor, but we are encouraged to work with specimens and species that can have certain logistical benefits. So for example, we as scientific thinkers appreciate, replication, sample size, and the logistics of caring for fish in a laboratory setting. Those things encourage the work with smaller fish. So a lot of the research that's out there is dominated by smaller fish species like stickleback or silver sides. And surprisingly enough, there needs to be more work with the larger game fish species that are sort of more recognizable to the everyday fish enthusiast. Now going back to, so there needs to be more work, more rigorous scientific work on that front when it comes to that statement that I said. But when I think about the East River fish, my thinking, and I say this all the time when I'm at the fishing clinics, is if you can catch fish consistently in the East River, you can catch fish anywhere. Because I've traveled elsewhere, and I don't want to take anything away from the beautiful areas that I fished across this country. But there's something about, maybe it's the New Yorker and me and just the pride of being in the city, but it's one thing if I can take, for example, someone fishing, and we're fishing, let's say in a bay, and we're targeting striped bass, and you're holding a rod, and I tell this person about an experience that they might encounter where, for example, a fish might come up and pick up your bait and spit it out immediately, and then do it again and again, as if it's checking to see if something is weird going on before they decide to commit to eat it. That happens a lot in the East River when we target fish, like when we go fishing for catch and release for striped bass and tautog. And so those are those experiences that I have growing up that make me really appreciate just how challenging but also rewarding it can be to interact with fish. And once you catch it, you release it and send it on its way, and just appreciate just the intelligence that they have.
Yeah, absolutely. I personally haven't done too much angling myself, but I think it like sort of like you were saying with the complex bottom habitat, they tend to have better spatial reasoning skills. We know that the New York area is very complex bottom habitat, so that definitely makes sense. Those are great stories. Okay, another question. Someone said, I saw that you work in sustainability. I would like to ask how we as New York students who are unable to donate can help with sustaining our home environment and New York.
That's a great question. My first comment to that is you can donate. You can donate time. You can donate knowledge. The one thing that I often think about when we think about fish in the East River is for most people, they don't get to see the fish unless you're participating in a hobby or an activity that draws the fish out of the water. And so having experiences like joining a program or participating in a community science event or going fishing, every single moment is an experience that can contribute and enhance the knowledge of others. Yes, resources would be nice, but that didn't limit me. I grew up just interested in fish, and here I am today, 20, 30 minutes sharing knowledge about our fish. And so don't ever underestimate the contributions you can have both intellectually, and if you strike it rich one day, definitely consider giving back because it helps too. But there is no shortage of need. And Roberta, we had a conversation about this before. When I was about 17, I attended a college course on marine biology, and the professor there shared with us this one line. He said, "The ocean needs as many friends as it can get. So don't ever feel like you can't contribute, because whatever you can contribute, the ocean needs." Okay, so the more the merrier.
That's great advice. It definitely stuck with me when we first had that conversation. It looks like we have quite a few friends of the ocean because we have a ton of awesome questions. So we're going to try to get to as many as we can. And if we don't get to your question, I'll put down an education email address for the park in the comment section. You can anyone can send questions there, and we'll see what we can do. So moving on, this next question. Fish have memory and ability to learn, as is evident from your work, but what about emotions? I keep hearing stories about large fish in public aquariums currently not doing well, maybe experiencing loneliness, while there are no visitors during due to COVID closures. And as an alternative, they're being given visual enrichment by being allowed to watch Netflix. So can you speak to that at all?
So that is an extremely important question. I'm not an expert when it comes to research on the emotional capacities of fish, but there is research out there. So in the field of psychology, if you get a chance to look up work on the mammalian amygdala, there is work on a possible amygdala analog in fishes. And so exploring sort of the connection and questions that relate to, for example, those things that were shared with regards to emotional impact. And one thought that comes to mind is if you look up if you look up a lot of behavioral research, my research focused primarily on food deprivation as a motivator. But there's a number of really cool research studies that show that social cues, so either in lieu of or in addition to food, having a separate compartment with other fish as a motivator to learn a maze is just as effective. And so the need to, especially certain species that are more social, the need to find other species and that being a motivator to solve a problem is something that there is research on. I would say that I don't have a definitive answer, but there is work out there with regards to thinking about conversations relating to pain, to suffering, and so just in terms in thinking about fish. So exploring those questions moving forward will be extremely important, but also realizing both the similarities and differences that the brains of different animals have is going to be really, really important conversations to consider. I did do some work related to rearing fish in different types of habitat. So there is this notion of environmental enrichment, so providing structural complexity in a setting that is, for example, in a fish that's held in captivity. And although the research that was very minimal, that there is work just exploring the capacity or the ability or the impact of having an enriched environment versus a non-enriched environment. So that question, I apologize as I don't have a more definitive answer, but certainly there is research out there and research to be done. And I look forward to seeing what future conversations we'll have about that topic.
Great. Yeah, thank you for touching on that. It is a complicated question. There's so much we don't know, but that's a really wonderful overview of that. So we'll have a couple questions. We have a couple questions from Lamont's program. So one of them is, how do eels know where they are going when they are migrating back to the Sargasso Sea from the Hudson River and from other areas?
So I'm a stickleback biologist, and I am going to put my colleague on the spot. So Chris Bowser, he is an eel expert.
Yeah.
And if he gets if he's watching, or if he gets to watch this, know that he is the first person I think about when it comes to eel questions. That is a great question. Just in thinking about migration in general and how different species do it. For example, there's work on salmonids and benthics, but eels, I mean, that's a whole other story that I won't try to attempt to answer, so I'll leave it there.
Okay, that sounds good. So we'll go to another one, which is sort of, you touched on this a little bit, I guess, but someone asked from Lamont's program, is there anything unique about Hudson River fish when compared to those in other aquatic environments in your experience?
So what's really special about the Hudson is, well, I guess in a sense, when you think about estuaries in general, year to year, it could be somewhat unpredictable what you find in different parts. Of course, every year there's always really cool, rare or often unseen catches. So one thing that one place to look in terms of seeing, for example, consistent catches, expected catches, and rare catches, I want to suggest looking up the Hudson River Almanac, and Tom Lake, who is a colleague of mine. He just maintains amazing records of what's found and historical accounts of what the Hudson has in store and what you find currently. In terms of what is different, I'm not quite sure how to answer that question. I think that fish have their tendencies, and it's a very specific question for me. Like, I would need to know maybe a specific species. But one thing I find is I've taken students fishing out in parks like Piermont Pier and Nyack Memorial Park, and I'm always surprised every year, especially in the fall, catching fish that you never expect to see because of a simple sort of relatively simplified understanding of what I think you should expect to find. So for example, in Piermont, one year we caught weakfish, we caught oyster toadfish, which I typically associated with being more marine. Now it could be that there was a brief period in time in which the water was more salt water than it was previously, but kind of coming to expect the unexpected is something that I appreciate about the Hudson. And this is the stories you hear. So again, such an excellent question. Not sure completely how to answer it in a definitive way. But the thing is, sure, the more eyes that are out there, the more cool things you'll find. And just looking at the almanac from week to week, you will see that. Yeah,
I definitely agree with you. The almanac, Tom Lake's work is incredible. We report to him our fish ecology survey results every week, and it's always a highlight of our week. So we have time for just a few more questions. So this is a bit of a different different one, different sort of frame of reference, which is, how do you juggle being a teacher, a research scientist, and having a family?
I don't. That's a really good question. How you kind of just do it, and I think it's really important to check that you're doing it okay. I will be honest. You know, there are times where one of those aspects tend to occupy more time than I would like it to, but in some cases you have to get the job done. You know, if there's a particular field season, you have to be out there. You know, you can plan ahead as much as you can, but at the same time, you know, there are times and places where sacrifices are made. Kind of the reality of the way it works. Having a loving wife, having a supportive family makes it that much easier and doable. But I think everyone's story, I think that's a great question. I think you should ask that to every researcher and educator, because I'd like to know today. I hope I'm doing it right. And at the end of the day, I do try to check and see if I am, because it's just one of those things. It's a life thing, you know. You hope that you're doing it right, and you have to, but hold yourself accountable and make sure and check in often. And yeah, that's
a good answer. That's a good answer. Yeah, it's a difficult balance, but you know, you're clearly doing it and doing it rather well. So.
I hope so.
So this is more of an angling question. What kind of bait seems to be the most effective when it comes to striped bass in particular? Is there one?
Oh man. Oh man. How much time do we have to answer the question?
We have, I think we have like 7 minutes left. Okay. And I think maybe if we have this one, and then we'll try to get one more question in after this one.
Okay. So
try to do two more.
It I mean, I would probably want to sit down with this angler and assess what tools they have. Like, for example, what types of rods and reels they have, what kind of experience they have. I mean, obviously there are these cliche terms like match the hatch, which basically indicates that, you know, fish are animals of habit. They tend to prefer and find and choose baits that are present. And so, for example, striped bass will eat eels in the fall. They will eat Atlantic menhaden in the spring, and silversides and sandhills in the summer. And so having baits, or even artificial baits that mimic those species, are important. You know, I can't tell you how many times early on fishing thinking, hey, you know, this bait will work, and not realizing, you know, you have to think like the fish. If you're swimming around and eating hamburgers all day, and all of a sudden there's this amazing steak there, it just seems out of place. And I'm pretty sure fish, especially the older ones, recognize that, and they recognize their patterns. And so I would say, if you're interested, definitely I encourage you to reach out to me, and I'd be happy to direct you towards meaningful experiences to answer that question, because I'm still trying to figure that out too, you know. The biggest fish I ever caught is small compared to those that I know who are much more proficient at angling than me. And that's what makes the hobby so fascinating and intriguing, is that there's always an opportunity to continue learning and learning from others. And so there's definitely like a kind of a real-time trial and error that takes place. And the fish let you know if it's working or not. You'll catch them, and it's pretty straightforward.
Good answer. And I guess I'll just last question, and I'm also going to try to respond to some of the other questions that I can respond to in the comments, and then I will be putting the email after that. But for a last question, Peter, we have this is coming from, but Someone is asking, how does a fishing clinic operate? What does that look like for someone who doesn't know, and how could they potentially get involved if they are interested in participating?
So I know that you could definitely answer part of that too. One thing that I've taken away from my over the years is that every community fishing clinic is different. And so, for example, when we thought about the Randall's Island Park Clinic, and we thought about the World's Fair Marina Clinic, and we thought about the Brooklyn Bridge Park Clinic, the identity, the community is different, and it's not always easy to predict either, because in some cases you'll have a lot of visitors coming, so it's not necessarily live in the neighborhood per se. And so in terms of getting involved, certainly looking up local programs, local environmental organizations that might host them, also looking the New York State Department of Environmental Conservation Region 2 website that lists the fishing clinics that are planned and have been approved. So there's definitely ways to find opportunities. If they're also for hire fishing operations to learn fishing, and paying to jump on a boat, for example. I did a lot of learning that way as well, and so supporting our local for-hire fishing operations, I strongly encourage that as well, because there's a long history, an amazing history of that across the generations in the city. When it comes to identifying the clinic that best works for you, other than kind of convenience, like getting to the location or knowing what you want, so if you want to spend a lot of time there with trying to do something on your own. There are so many variables that go into angling, and there's so many variables that go into what people want out of fishing. And one thing that I try to do with my students is it's really exciting when you're taking someone who is new to fishing, because they have no idea what angling is like. And I always tell them, you're going to discover the type of angler that you're going to become. If it's a bait fishing, if it's lure fishing, if it's a springtime or just the weekend. And you don't know that yet, but as you fish, you're going to discover that you have certain tendencies and preferences, and watching that develop and being available, or encouraging even my friends to, if I'm not so much for example, a largemouth bass lure fisherman, I have friends that are, and just knowing that there are all of these paths that one can find their space. And you have to understand too, like for me growing up, fishing was in many ways an escape just from the pressures of being in high school and college. And so having that hobby and just spending the time and having a heart that's committed to get good at something, you'll learn something that you will then teach others how to do. And so I would say definitely, if you're just starting out, consider clinics and consider for-hire fishing operations. And as you figure out the type of angler you will become, you can ask me, you can ask. There's so many amazing social media groups out there too that really encourage the hobby, and and just be prepared. Be prepared to have days where you're not going to catch fish. You learn most on those days, and the teacher in me will tell you, document everything. Keep a logbook, because you don't want to repeat the same mistakes. And as you get older, you have less and less time to go fishing. And so with that less time, you want to make sure that the time that you do have, you use it wisely and efficiently and practically. And so document, try not to repeat mistakes, but also be open to learning the kind of learning and discovering the kind of experience that you want out of angling or seining, or any other type of fish collection, catch and release type of opportunity that's out there. There's so many in the city.
Awesome. Thank you, Peter. I think that is just about all the time we have for questions today. So I am putting the Parks Education email in the chat. If you did not get your questions answered, you can email us, and we'll try to get those to Peter so that we can answer your questions. So thank you again, Peter. This was wonderful. I hope everyone watching enjoyed it as much as I did. We're very much looking forward to continuing to collaborate with you, both virtually and hopefully in person eventually. And thank you everyone for attending.
Thank you so much, Roberta, and thank you so much just for the privilege to be here and to do this. It was so much fun.
Good. I'm glad. Thank you so much, everyone. All right. Until next time, you can tune in tomorrow for Live from the Field water quality at 2 p.m. We're going to be doing basic testing. And the next Ask a Scientist will be in early August and will be a diversity in STEM panel. So thank you, everyone. See you soon.